Welcome back to the Business of Biotech. I'm your host, Matt Pillar, Chief Editor at Bioprocess Online, and I'm glad you tuned in. So to set the stage for today's conversation, I'd like you to ponder a few questions. What if you had a near real-time in vivo view of your molecule's efficacy while it was doing its work in the patient's body? How would this view inform biologics development? How would it expedite clinical trials? How would it change your approach to patient monitoring and reporting? While it might sound a little bit sci-fi, there's a company called Glympse Bio, an MIT spin-out formed up in 2015 that's currently in the clinic with a portfolio of bioengineered, tunable sensors designed for in vivo diagnostic and prognostic purposes. These biosensors are custom built to monitor the status of a range of protease-mediated diseases and their response to therapeutics. The company's leader brought nearly 2 decades of big pharma leadership with her when she took the reins at Glympse in 2018. After heading up the R&D and regulatory policy initiatives as SVP at the trade group PHRMA, she had held various VP roles at Merck and Bristol Myers Squibb, most recently as VP, head of R&D strategy and planning across the whole portfolio of clinical products, clinical candidates, that is, at BMS. Her name is Dr. Caroline Loew, and I'm thrilled that she's here with me today. Dr. Loew, welcome to the show.
Thank you, Matt. It's a delight to be here. Thank you so much for inviting me today.
Oh, it's my pleasure. It's a super interesting conversation we're going to have. I'm excited about it. Did I get the brief overview of your background correctly?
You definitely did. I'm excited to talk about it a bit more with you today.
Awesome. So, as I noted, as I was kind of setting this up and commenting on your background, you've got some big pharma experience, a bunch of it, and significant roles, right? I'm always intrigued when I talk with CEOs of biotechs who leave big roles in big pharma to start clinical stage companies or work for clinical stage companies. So I'm just hoping we can start there and get a little bit of background, Dr. Loew, on why you made that choice. Why'd you leap into a clinical stage biotech from big pharma?
So, it was something that I had always wanted to do. I had I had this kind of view of my career trajectory from very early on that I wanted to run a smaller biotech startup. And I don't know that I specifically know exactly what that was going to look like, but I had some view that I wanted to do that I wanted to build something. And I actually had very methodically kind of planned my career with that aim in mind. And, I had thought about, a number of different things. And my view had been that to be able to be successful kind of in that biotech arena, you really needed to understand, what it took to successfully develop a therapeutic or to bring a product into market. And so I sort of systematically set about building my career in large pharma, gaining those experiences, right? So I was very fortunate to successfully lead a drug to market on the drug development side. I then had a deep experience in the regulatory arena. I built and led a global market access organization, and then ran a large commercial business unit, a $7.5 billion commercial business unit, and then finally, as you pointed out, sort of capped all of that experience off with a very broad role at Bristol Myers Squibb, sort of number 2 in the R&D organization, responsible for portfolio strategy and operations. And what that did was kind of bring all of those experiences together and say, what does it do? What do you need to do to sort of successfully, prosecute a portfolio? And for me, that was kind of the capstone experience. It was really about saying, did I feel I kind of had everything together? Had I learned everything? And towards the end of that experience, I decided that, it was sort of time. It was time to make that leap and, think about what next. And, it was an interesting transition. I spent about a year thinking about it, and not just thinking about it, talking to a lot of people who were, currently CEOs of biotech companies, leaders in the VC community, trying to understand sort of what it was that I really wanted to do. And it was an interesting journey because I actually had planned to do something that was quite akin to what I was doing previously, but in biotech land. So essentially, running a well-established, clinical stage company with assets in the clinic, maybe, 100 people, that kind of size company. Bearing in mind I was coming from, a, leadership role in a, 6,000 person R&D organization.
Yeah.
And with, many, tens of clinical stage assets. And instead, what I realized through that journey was that what I loved doing more than anything was building something, sort of this process of creation. And, I love building something. I love solving really hard problems. That's what really drives me. I sort of like the idea of the blank sheet of paper. And, like many in our industry, right, have a passion for the power of kind of the transformational power of science in our business, right? That how that can really, you can really bring that to bear to solve, difficult problems in all domains. And in the end, difficult problems that make a difference for patients. And, I've had a belief, and I've pulled that through my career, that when you bring all of those things together and you bring integrated teams, you can make a huge difference in complicated problems. And so for me, it became about finding sort of the really hard problem that I was passionate about with some really great science and being able to build something. And so, it was sort of the moment was right. I felt like I had built the skill set necessary to build a successful company. And then it was just about finding that science that was, ready to solve what, I felt was a really complicated problem. And it's if you talk to a lot of leaders, who are fortunate to be in the situation that I'm in, I think a lot of them talk about this experience, right? Sort of finding something that they're incredibly passionate about and having the opportunity to build. And it's been a wonderful journey for sure. And, hopefully many more years to come now.
Yeah. And I mean, it's certainly, it's an interesting company, where I do typically interview former big pharma, current biotech CEOs. They're working very specifically in maybe a therapeutic development. This is a super interesting and, pioneering technology. Tell me how you came to learn about it. I understand that it was a spin-off of MIT, a product of, I'm probably going to butcher her name, but Dr. Sangeeta Bhatia.
Bhatia, yeah, that's right. That's right.
Yeah.
And sorry, go ahead.
Well, I was just curious that, at what point and how did you come to meet perhaps Dr. Bhatia and her technology and decide that this was something shortly after the company was founded in 2015, you joined it in 2018. Just give us just a little flavor of the genesis there.
Yeah. And so this was really interesting because, I have a wholly therapeutic background and, my experience had been, very much in innovative therapeutics, and most recently at Merck and Bristol Myers Squibb, right on the cutting edge of therapeutic development. And I had wholly expected to run a therapeutic company.
Right.
Diagnostics was just not on my radar. But very interestingly, one of the biggest issues that I had grappled with in the portfolio that I was managing in BMS was an issue that is, very straightforward. It's one that is experienced by every, person in therapeutics, whether they are, working on a single asset or working on a portfolio. And it's sort of this question of, how do you discriminate between the winners and losers? How do you tell whether, who is going to respond to your treatment? How do you have target engagement? And when you scale up to a portfolio level, these become, pretty significant issues. And, we see time and time again, substantial failures because these issues haven't been well discriminated. And I can tell you that in our oncology portfolio at BMS, we were grappling with this. I really sort of started to question when, when was our essentially our diagnostic capability, our ability to query response at the cellular level, going to catch up with sort of the therapeutic advances that we were seeing. We were seeing a lot more, a big advancement across, targets therapeutically, a big evolution again in small molecules as well as large molecules. Lots of different modalities were coming forward, and yet we were still not really able to understand response well. Mm-hmm. And it appeared to be, it was really frustrating our ability to advance therapeutics, and not just in oncology, but in other disease areas as well. NASH, where we're developing our first diagnostic, was a classic case where we're stuck on biopsy confirmed endpoints, in the painful, difficult biopsy that's very heterogeneous and very capricious to work with in the clinic, and very difficult for the patient, candidly. So the drug development is littered with this problem.
Yeah.
So anyhow, so an issue that I had seen. So, I'm thinking about this question. I'm getting to the point now of starting to quite seriously explore very specific technologies. I was only looking at therapeutic companies. So everything I was looking at was therapeutics.
Right.
And I was talking to Polaris, the venture firm, and one of the partners I was talking to there, actually Amy Schulman, who is on our board at Glympse. I was talking to her about a therapeutic company, and she said to me, "You should also take a look at this company, Glympse. They have really cool technology. I think you'll love it." And as soon as I saw this technology, I was like, oh my goodness, this technology has the potential to solve exactly the issue I just described to you. Yeah. And it was like a light bulb went off for me. And until that point, if you had told me, go and look at a diagnostic company, I would have said, I'm not a diagnostic person. Diagnostic is not my world. This is not something that I'm interested in. But seeing what the potential was for this technology, and with its platform capability, the fact that it's querying fundamental biological response to both address this issue, but then fast forward to become a broadly available diagnostic, mm-hmm. was just game changing for me. And I'm like, to be able to go and be part of evolving that was quite incredible. So very quickly, I met Sangeeta and heard about how she had really sort of nurtured this from the very earliest. The very earliest observations, and that was over the course of, close to 15 years. It was just incredible to see that journey and the power of the data that they had. They had incredible depth of data. And so this wasn't, like a lot of technologies where you see it and it's sort of like, there's a Nature paper that was published 2 years ago and you're like, oh, no, you've got, 10 years of discovery work. This was an extremely well-established technology with a tremendous breadth of data behind it. And so it gave me a lot of confidence that this was something I could really propel forward. I wasn't going to be, sort of stuck in the mire of, 10 years of preclinical and then clinical work. I could really accelerate this. And that was sort of they were kind of at a tipping point. And I was very fortunate. I, got the opportunity to come in and build the company, and, it's been great. I mean, we completed some, incredible preclinical studies, propelled into the clinic, and moving to late stage clinical development. We can talk about that more later. But, this sort of journey has been phenomenal, absolutely phenomenal.
For sure, yeah. And it's afforded you the opportunity, though it is a diagnostic company, it's afforded you the opportunity to further the important work of the therapeutic companies, perhaps the very therapeutic companies like that, which you thought you would progress to after you left Big Pharma. I mean, you didn't walk away from it. You're playing an integral role moving forward. So I think that's very cool. So, so we've, this has been quite a preamble, and I'm sure our listeners are sitting here going, okay, so let's talk about the technology. What is this? What is this? What are we talking about here? So
that's right. Well, you did a pretty good job introducing it, but let me give you a little bit more of a vignette. So what we can do is, it is very cool, and we can detect real-time biological changes at the site of disease, and we do that using basically these activity-based sensors that are bioengineered, and they're tunable to any protease-mediated disease. And so essentially what they do is they go in and they query disease activity and they report it out. So this gives us, clinically actionable real-time disease data. So we can detect disease, we can detect response to disease. And so this concept is extremely simple. It's extremely elegant in its sort of approach, and it's filling this a void essentially in the world of biomarkers, where we're synthetically creating the biomarker, and we can tune it to any disease. So these are bioengineered sensors. We tune them to each disease state, and how it works is pretty straightforward. In this first presentation, the form that we're working right now on clinically, they're injected. They localize to the disease state. They localize to the tissue that we target. So for NASH, they localize to the liver. And they just measure the activity of the proteases that are dysregulated in that disease. And then in an hour, they're excreted in the urine, and then we detect them using standard lab approaches. We measure them using a classifier that we develop. That classifier is specific to the disease state. And then they're reported out as clinically actionable results. So in the case of NASH, for example, we can do two things. We can both stage the disease, so we can tell how severe the disease is with a lot of fidelity, and we appear to be able to detect it with far more fidelity than any other diagnostic that's available today. And the second thing we can do, which is particularly valuable, is that we can tell whether disease is progressing or regressing. So imagine now you're being treated. We can tell, are you responding to treatment? How well you're responding to treatment. So sort of the rate of progression of your disease. And then the really cool thing, sort of in the future, fast forward, I mean, this is kind of like, a little bit downstream, but because we're now measuring kind of native biology, this protease biology hasn't been mapped before. Because we're measuring this native biology, as we aggregate this protease data together with other, clinical measures, we will, we'll actually be able to, we'll sort of create a data engine, which will allow us to do two things. We can first increase the fidelity of the analysis that we develop, so sort of the precision, but which allows us to, powers more discovery if you want. But we would imagine being able to make population-based level assessments. So we might be able to say, based on an aggregate body of data, we might be able to look at a patient's analysis and say, based on your profile, you're likely to respond to drug X. Based on your profile, you're likely to respond to drug Y. And that's just based on aggregate patient analysis. And so, I think we envisage these tools being very, very powerful over time.
The Business of Biotech is brought to you in partnership with Cytiva. Together, we're committed to helping the leaders of new and emerging biopharma companies navigate the financial, organizational, human resources, and regulatory waters you'll encounter on your way from discovery to the clinic and beyond. Check out a host of useful resources for biotech leaders at Cytiva's Emerging Biotech Accelerator at cytivalifesciences.com/emerging biotech. That's CYTIVA lifesciences.com/emerging biotech. Very powerful and very applicable to, I guess, displaying broad applicability. So you develop this technology, this tool, and then, obviously you've got to go, okay, so where does it fit? Who can benefit from this tool? And one of the reasons I wanted to talk with you on the show, Dr. Loew, is because, I'm Matt Pillar, master of the obvious. I thought, man, how powerful a tool could this be to clinical stage biopharmaceutical companies who are measuring the impact of their therapeutics in clinical studies. I imagine that's but one element of your sort of marketability for Glympse Bio. Mm-hmm. So take us through that. You know what my ulterior motive in having you on the show was, and I'm hoping that we can talk about how your solution can benefit biotherapeutic companies in the clinic. But let's start sort of at a higher level than that, and walk us through, I guess, the applications that you see in the life sciences for your technology.
Yeah. So ultimately, we're a diagnostic company. So we want to develop a marketed diagnostic, and we see that marketed diagnostic in most cases doing two things, right? Can identify disease, can identify response to treatment. And interestingly, when you look in all the diagnostics, you don't often see those two things together, right? You see one or the other. Maybe you can't see the breadth of disease. The other thing is that you see in a lot of diseases, there's kind of this diagnostic void, right? That people go on these terrible, multi-month, sometimes year-long diagnostic journeys. And so we're focusing our technology into those areas where just the diagnostic journey is complex. Because we can tune the technology to the protease mediation of disease, we're able to probably deconvolute complex diagnostic journeys. So that's helpful, right? So that's, in the end, we're a diagnostic company. And, that's the big opportunity. Yeah. But to your point, kind of the world that I came from, the world that a lot of our team comes from, we see enormous value at this intersection with therapeutics, right? And that spans the entire drug discovery and development continuum. And, we already have collaborations in that space, and we see enormous value in that space. So you can back up right into drug discovery, where, we can have collaborations to support, target engagement. You can take this is the beauty of this technology. You can take exactly the same diagnostic directly into early clinical studies. Mm-hmm. And there you can tell whether you have, early efficacy response. The same diagnostic can then be taken into full clinical studies as, for patient identification and for, surrogate endpoints, again, for early efficacy. Can then be used as part of your, approval packages, and also to support reimbursement. And so you sort of have this end-to-end use. So then, yeah. To your point, what does it become useful for, right? So you go through this process of validating it, right? It helps you in the, what I would call what I use this term carefully, the sort of the translational research space, where you're trying to identify whether you have an asset that you can progress, into later stage clinical development. It can really help you identify whether you have efficacy response, and that there's a very significant failure rate in that area right now. Still, for pharmaceutical companies, it's very hard to cleanly understand whether you have response. Yeah, and so technologies that can help deconvolute that, because it can either speed that up or increase the probability of success. So speed the decision making, increase the probability of success, right? So make sure that you make better discrimination between the winners and the losers, are very valuable. And you just want to think about it in terms of time, right? If you can shave, a month or 2 months or 3 months off that decision making, right? Or if you can, make a better decision on one asset, all of that becomes very valuable. Probably for the average large pharma company, they're spending, $300 to $500 million a year in that portfolio. And so anything that you can do to help, improve the decision making there and the speed of decision making is tremendously valuable. And so we see a lot of opportunity there. And in part because from some of our data both in NASH and in oncology, we appear to be seeing response, or if there's no response or because of absence, we appear to be seeing that far earlier than any other diagnostic tools or endpoints that are used. And so, we're still in the early stages of validating our technology, but, if that continues to play out, right, if it plays out in the studies that we have currently in the clinic, then that would be tremendously powerful. When you can increase the probability, you can improve your decision making, you can improve the speed of your decision making. Those are all good things, and that's been sort of core part of the past probably 10 years of evolution in the drug development space. Is how can you bring better tools to those processes? Sure.
Yeah. How early is early? So you're looking at clinical stage, even preclinical discovery stage companies. So let me ask you this flatly: does the technology work in animal studies?
Mm-hmm. Yes.
Yeah.
Yeah.
Okay.
Yeah.
Yeah.
Yeah, absolutely. And so, very interestingly, we use human proteases, and and those human proteases work in animals. And what we're seeing is we believe that there's that there's been this very strong evolutionary conservation of protease biology, and so we sort of now appear to have this advantage that we will derive a biological panel from human RNA-seq data for any disease, whatever that happens to be, and we'll say we'll have panel X. We can use panel X in across a multitude of animal models. It will work as well in those animal models, or appears to work as well in those animal models across a range of different animal models as it appears to work in humans. Now we've got that now. We've got that experience in NASH. We're seeing similarly across animal models in oncology. We haven't gone into humans yet in oncology, but oncology is much more capricious, so I'm cautious there. But there does appear we do appear to have some advantage of this potentially biological conservation of protease biology across mammalian species that is helping us in the development of our technology.
Yeah. So you've mentioned NASH, you've mentioned oncology.
Mm-hmm.
Our audience is primarily new and emerging biopharma, clinical
mm-hmm.
stage and early stage biopharma.
Mm-hmm.
What types of companies among our audience would be, by however you would want to slice and dice it, by size, by modality, by target? Who's a good fit for Glympse?
Yeah. So we think about it largely in terms of therapeutic areas.
Yeah.
NASH obviously is our later stage clinical target. We would generalize NASH as fibrotic diseases. And so that's a much bigger basket. And there's a lot of common protease mediation across many fibrotic diseases. And so our panel, we believe, has a lot of, generalized applicability. And so IPF might be another example of that, but there are, other related fibrotic diseases. So fibrotic diseases generally, oncology, and that's sort of an adjacent, disease with some, related, related protease mediation. But then also in general that, other protease mediated diseases as well. And there are many therapeutic companies who are focused on, protease mediated therapeutics. So, that's sort of the universe that we've been in. We've also we've also got some data in infectious disease, other liver related diseases. So there are a lot of adjacencies that we have looked at. So we have preclinical data. I think NASH, fibrosis, liver, and oncology, infectious diseases are sort of the areas that we have concentrated a lot of our research. Our main programmatic areas right now, most advanced in NASH and oncology.
Yeah. Okay. And I mean, we talked about, we talked about stage. When does it make sense for a biopharma company who's interested in your technology to engage? Is it as early as possible kind of thing to extract the most benefit?
Yeah, I think the most productive partnerships are ones where, for a company like ours, where a company sort of travels the sort of the journey with us, goes from preclinical to clinical.
Yeah.
And that's because there's sort of a mutual learning, right? That you can generally learn from each other. That said, our NASH technology is pretty advanced now. And so in general, it wouldn't make sense to kind of go back and recapitulate what is now all of preclinical data, right? You'd want to be in, later stage clinical development to take advantage of the technology. There's no point doing that. Oncology, our program is earlier. It's, close to being in the clinic. And so there, I think, that's a better example of a technology that's, or an indication that's, later stage preclinical, about to go into the clinic. That will be a good use case for that. But, my when I talk about this and I think about it, I think companies like Gilead's traveled with us for a number of years now. And I think we have both benefited tremendously from the arc of that relationship. We've both learned a lot. And, I think it's been, a very beneficial, highly collaborative relationship.
Yeah. So we've talked, you've alluded to the value that I just mentioned, at what point at what point should a company engage you to extract the most value? And we've alluded to that value being efficiency, recognition of efficacy or not, right? Like fail fast, fail cheap, right? Knowing who to knock out. What other, are we missing any other sort of value statements around, I don't know, maybe quantifiable or even, even qualitative kind of statements around what the value is to, a new and emerging biopharma in the tech?
Yeah, one of the interesting things that, we see particularly in the NASH space, and I think it's a general issue, and, I had a lot of experience. When I was at Merck, I spent a lot of time in the payer universe, leading the market access organization. And, you look at the NASH market, it's sort of this big emerging therapeutic marketplace that the payers rightly, I think, have a lot of question about, right? It's, potentially going to be a very, expensive, untapped, market for them. A lot of, patients potentially going to be, open to, treatment who weren't previously on therapeutics. And I think they have a lot of question about that, rightly so. And, who should be treated, what's their response going to be? It's sort of, it's the same questions that came up when osteoporosis first, evolved as a market, for example, or, the type 2, the type 2 diabetes, market really opened out. You've seen this again when large therapeutic markets have opened with new therapeutics. And one of the values that we can really help with in that case is sort of twofold. The first is we can help identify the treatable, right? The patients who should be treated. And in this case, in NASH, because there's a disease of fidelity, right? There are patients who have later stage disease who are definitely going to be eligible for some of the new therapeutics that are coming into the market. And the current diagnostics don't really point to that extremely well. I think we should, with high fidelity, be able to identify those patients. That's the objective of our diagnostic. Yeah. The second thing is, and this is the most important thing, and I learned this time and time again from my payer experiences, we should be able to identify with our diagnostic whether patients are responding. And, as a patient, you don't want to be sitting on a treatment if they're not responding, right? It's the last thing you want. You want to be moving on to the right therapeutic. And I think that's the perspective of every pharma company too, right? They want patients to be treated to target. And that's the thing that we can really help with. And so for me, that's a big piece of this, which is supporting the access equation, right? I think the payers do want patients to be treated if they should be treated, but they want the appropriate patients to be treated. That's the same thing the pharma companies want. Well, there's sort of a link in the middle, which is the diagnostic that enables that. And that's the piece that we can help with. And then, sort of underpinning that, we can help them with, sort of natural history data and the like that can start to demonstrate, are there subparts of the population that are potentially fast progressors? How do you identify those? There's the things in a NASH market, for example, that are just not understood now. I think that there's better data, for example, in the, in the oncology market and in many cancers than there is in NASH. We would hope that our diagnostic can help to unpick that a bit and provide the fidelity that's necessary in the market.
Yeah. And I certainly don't want to give short shrift to the point you made earlier around, down the road population level metrics around response. I think, again, that in and of itself, there will be multiple applications for that information, that knowledge, that technology. So very cool. We're running a little bit short on time here, Dr. Loew, but I want to make sure that we have a little conversation about your pipeline. And in the context of a pipeline update, I'm curious about your regulatory experiences. So maybe we can kind of pack these two questions into one. Give us an update on where you are in the pipeline, and maybe just color it with a little bit of commentary around, I guess, one, your regulatory experience, given that this diagnostic tool is a relatively, if not entirely, new technology that perhaps regulators haven't seen before, right? So that's always interesting. And two, just around your experiences on the regulatory front in the age of COVID, whether it's been a hindrance, a disruptor, or if in fact it's accelerated your regulatory dealings. Boy, I just packed a lot of questions.
There were a lot of questions there. So we're entering late stage clinical development for our NASH diagnostic, and so that's sort of our next big step. And our pathway is a drug device combination. So we sort of the sensor combination, right? The biomarker combination is a drug that is sort of administered in a device, right? There's a syringe that gets administered. And so the initial pathway of the development which is the first in human that we did, is really kind of classic drug development. There's nothing much different about it. As you move into kind of a later stage, there are elements that are drug-like, and there are elements that are really, diagnostic-like. So we've been very thoughtful about our regulatory pathway. We engaged with FDA very early on to get this designation, drug device combination. It's CDRH led, but we have CDR at the table in all of our engagement with FDA. And they've been highly collaborative with us. So they're very focused on sort of the advancements of new diagnostic technologies. And we have found, in fact, the duality of that guidance from FDA to be extremely helpful, and also very helpful in the context of us, doing a lot of our development with pharma companies as well, or with Gilead in this instance. So we found this to be highly collaborative, very good guidance and input. It's been expeditious. We haven't had any delays. And in fact, during COVID, it was interesting. We were just going through regular guidance processes. We were, they issued sort of notifications to sponsors, broadly indicating that they were likely to be delayed. We personally experienced no delays at all. So we were very fortunate. And so I give them credit. They've been, completely overwhelmed, and I know this has been a great burden for them. They've been dealing with, everything that everyone else has, as well as I think just a huge inundation of, COVID-related, regulatory applications. But for us, we had no delays at all, so we've been very fortunate.
Excellent. Well, that's great news, and I thank you for the update and the education. We were commenting before we started to record that you and I, it's been, I think, about a year since we last talked.
It was a year. It was odd that it was about a year, right?
It's been a long time, but
a long time.
I just really appreciate the education when I spend time with you, Dr. Loew. It's fascinating. Not just the science, but your experience and what you bring to the table in terms of that experience that you can share with our listeners. So thanks a lot for joining us.
Thank you. It was great to catch up, and I really appreciated the time today. Thank you.
Yeah, we'll keep track of the company and hopefully get a chance to revisit where you guys are down the road.
I would love that. Thank you so much.
So that's Dr. Caroline Loew. I'm Matt Pillar, and this is the Business of Biotech. We're produced by Bioprocess Online in partnership with Cytiva, a company that's as dedicated to serving the leaders of new and emerging biopharma as we are. Check Cytiva out at cytivalifesciences.com/emerging biotech. Check us out at bioprocessonline.com, where I'd be ecstatic if you'd subscribe to my newsletter. And finally, if you like what you heard here today, subscribe to the pod and give us 5 stars. Thanks for listening.